right. Well, first of all, thank you all so much for coming out today to learn more about my dissertation work, which focused on striped skunk ecology across an urban-rural gradient in Southern Illinois. There we go. Okay, so first, just a brief overview of what my dissertation entailed. I had 4 chapters: winter denning behavior of striped skunks and interspecific activity at their dens, pathogen prevalence, spatial ecology, and then survival and cause-specific mortality. Now, for the sake of time today, I'm going to focus on the first 3 chapters, but the survival and cause-specific mortality results that I saw were pretty much in line with what had been reported in the literature for other striped skunk populations. And of course, if you have any questions about any of that, feel free to ask them at the end or get a hold of me afterwards. So with that, let's go ahead and dive into just some background information. So striped skunks are extremely widely distributed across most of North America. They're generalist mesocarnivores, so they're found, across most of North America in all of these different habitats where they eat a variety of different foods. However, they're classified as mesocarnivores. They're a medium-sized carnivore, and they eat about 50 to 70% meat. For striped skunks, that's primarily insects, but they also will feed on small mammals and carrion and really anything else that they can find, truly. They are an urban adapted species, so they regularly just sort of integrate into urban areas. And they're also host to many pathogens throughout their range. And in fact, disease and poor body condition are significant causes of mortality in striped skunk populations, including this one. So mesocarnivores and urbanization. Human activity in general has caused a large decline in large carnivores. Think of wolves and cougars. And subsequently, we've seen an increase in mesocarnivores. These are, again, our smaller sized carnivores that are eating 50 to 70% meat, but have a variety of different foods in their diet. They're also highly diverse. There are some mesocarnivores that are extreme specialists, while others are generalists. Mesocarnivores are often more abundant as well. So this is interesting, especially in conjunction with urbanization, because generalists with broad dietary and habitat preferences tend to be the types of species that can really benefit from urbanization and really take advantage of the situation of urbanization. And so mesocarnivores, really generally a lot of them fit into this description of species that could probably adapt to and benefit from urbanization. Now, throughout my talk today, you'll notice sort of this direction of, or this tie that's underlying it all, besides the striped skunk aspect, and that's this One Health approach. So this is the idea that pathogens infect humans and domestic animals and wildlife, and one single pathogen can infect species from all of these categories. In recent history, we've seen an increase in the emergence of zoonotic diseases from wildlife. Those are pathogens that are coming from wildlife and causing disease in humans. And this is, of course, a human health concern, but also the prevalence of diseases in wildlife populations is also a conservation concern, especially when we consider that this increase in emergence is often associated with habitat fragmentation and destruction, and humans' role in spreading pathogens around. So overall, the One Health approach is an approach that recognizes that human health and animal health and environmental health are all interrelated at both local and regional and global scales. These are all sort of connected to each other and impacting each other. And there are also a lot of other angles that come into this as well, outside of just the health aspect of things, such as biodiversity and anthropology and economics. So it's really just this idea that in order for human health to be a priority, we also need to consider all of these other aspects that relate. And so with that, let's go ahead and jump into my first chapter, which again focused on winter denning behavior and interspecific activity at striped skunk dens. So striped skunks exhibit variable winter denning behaviors. They occur across this super large range that has really different climatic conditions. But in areas where they're experiencing a harsh winter, they have this period of food scarcity that they're enduring. And so they typically participate in extended denning, where they're going into a den and they're staying in a den for multiple days. Sort of think of hibernation. They're not true hibernators, but it's a similar situation. And they also participate in communal denning, or huddling, where there's more than one striped skunk that is occupying a den at a single time, and they just sort of huddle together to preserve body heat. This is also important because striped skunks use denning structures that are similar to other sympatric species, similar to other species that are found in the same areas, such as raccoons and Virginia opossums and domestic cats, and the list goes on. So to tie this all back to human impact and human modification of the landscape, humans change landscapes and they also provide supplemental resources. So in this photo over here under these stairs, this is a striped skunk den in Carbondale. Humans provide these artificial shelter resources, sheds, barns, the list goes on. They also provide food resources and cat food and bird food and scraps left out for whatever might eat them. It seems innocent, but studies have shown that brown bear hibernation in Slovenia is shortened in the presence of humans and human activity and human foods. So because they have this food source all winter, they don't need to hibernate for nearly as long. So then you have brown bears active for longer periods of time during the year, and increases in human bear encounters and complications there. So there have been no studies that have investigated striped skunk winter denning behavior in the lower Midwest, and no studies have examined differences in striped skunk winter denning behavior across an urban-rural gradient. So moving from a more urban area through agriculture, through exurban development and suburbs, all the way down to a national forest. And thus the objectives of this study were to examine striped skunk winter denning behavior from November to February of 2018 to 2021. Specifically, I focused on the duration of striped skunk inactivity at their dens, the number of dens that an individual was using. I tested for effects of human modification on the landscape on the number of dens that striped skunks used, and also tested for the influence of weather, landscape features, and human modification on denning duration. I also used camera traps to document interspecific, so striped skunks and other species, and intraspecific, multiple striped skunks, activity at striped skunk dens, and tested for correlations in this activity. So I expected extended and communal denning due to the metabolic benefits in an area that sometimes drops below freezing and sees a little bit of snow. I expected that skunks would use multiple dens during the winter. To put that in context, during non-winter season, striped skunks typically are using a different den each day. So it seemed appropriate that they might use more than one over the entire winter period. I expected that warmer weather, increased precipitation, which is normally rain in Southern Illinois, and human modification would decrease the denning duration, shorten that amount of time, because of an increased access to resources. I expected that water would be a limiting resource, and that would promote longer denning durations closer to water sources. And I expected fewer dens in areas of increased human modification just because of the sheer number of resources in a smaller area. As far as interspecific activity, I expected that there would be interspecific activity at den sites, just due to the number of sympatric species that use similar dens. But I didn't expect any correlations between striped skunk activity and other species activity, just because striped skunks generally are not very territorial or social. So for this part of the study, I captured striped skunks and radio tracked them during the winter at least 4 times a week. I set camera traps outside of dens where striped skunks spent at least 3 consecutive days. And then I used the information from tracking these skunks to build 95% kernel utilization distribution home ranges, which I'll talk a little bit more in detail about that later in the presentation. And I also calculated the denning durations, how long a striped skunk spent in a den, and then the mean temperature and precipitation that occurred during that time spent in the den. I also incorporated some spatial covariates. So I calculated the mean human modification score on a scale of 0 to 1 within a 1 kilometer buffer around each den. That approximates an average home range for an individual striped skunk. And then I also calculated the mean human modification score within each striped skunk's home range. This is sort of what that looks like. Human modifications of 0 are areas with very little human modification, very little human activity. Think of contiguous forests. Human modifications of 1 are very high levels of human modification. This is, a city setting. And then we might see out here on the outskirts some agriculture, higher road densities, higher night light pollution, navigable waterways and railroads, just signs of human activity. I also calculated the distance from each den site to the nearest road and stream or shoreline. And while we're talking about all of this, I just wanted to note that I was tracking the striped skunks, so I knew that they were using the dens that I was tracking them to. But the animals that were captured in photos outside of these dens, I had no way of confirming that they were actually using the dens and not just, sticking their heads in and leaving. So because of this, in my analyses, I considered any animal that was captured outside of a den site active outside of the den. And so you will hear me throughout the presentation here discussing activity outside of the den and not denning together. Just a note. For my analyses, I used a Poisson regression to test for the effect of mean human modification score within a home range on the number of dens that an individual used. I used generalized linear mixed effect Poisson models to test for the effects of spatial and weather covariates on denning duration. And then I also tested for correlations between striped skunk activity at a den and other species activity at a den site. And I found that individuals used between 3 and 21 dens during a single winter season, with an average of about 9 dens used per winter. There was no significant effect of human modification of the landscape on the number of dens that striped skunks were using either. In this study, which I've marked here with a star, I saw up to 59 days of consecutive denning. So a skunk went into a den and stayed at that den for 59 days. I also detected some communal denning where we had more than 1 skunk at a den at a single time. This sort of fits into this pattern, this latitudinal gradient that we see in North America, where in Canada with harsh winters, we see skunks denning for up to 150 days. And also some communal denning. But then down here in Texas, we see no extended denning, no communal denning. And that's probably just due to the change in winter conditions. It is worth noting over here with Thymer, there's sort of a break in this pattern with 92 days of extended denning all the way down here. But the authors of this study correlate this with a higher elevation. And so a harsher winter climate because their study took place at a higher elevation. As far as denning duration, I found I had one supported model that included human modification, the distance to streams, and the mean temperature during denning. And when we take a look at that model and break it down, we see a significant negative relationship between the duration of denning at a den and each of these three covariates. So as human modification increased, denning duration decreased. As the distance to streams increased, denning duration decreased. And as the temperature increased, denning duration decreased. I detected 11 different mammalian species outside of striped skunk dens. This is sort of worth noting here. We have 2 skunks captured in this photo outside of this den, and neither one of them are the collared skunk that was being tracked. It's also interesting here, this photo was captured on February 3rd, this photo was captured on February 5th, and this photo was captured on February 6th, all at the same den site, but within a very short period of time. So not all there at the same day or same time, but really stacked up back to back. And we're going to come back to that in a little bit here. As far as interspecific activity goes, I did detect a significant correlation between striped skunk activity and Virginia opossum activity. And then I also detected a significant correlation between bobcat activity and chipmunk activity, which that one's probably pretty easily explained by a well-known predator-prey relationship. When we take a look at when photos were being captured during the winter, we also saw this interesting trend. We had about the same number of cameras running throughout the seasons, and yet we see a higher density of photos here in the second half of the winter season, despite fluctuations in temperature through all 3 years. I suspect that this is probably correlated with the onset of breeding season in many of these mesocarnivores. And so overall, I detected, or my study fit in with this latitudinal gradient in the duration of extended denning and also in communal denning, but we do know that elevation may impact this. This is also noteworthy because typically striped skunks lose about 50% of their body weight during the winter with all of this food scarcity and extended denning. They really come out of winter in a very poor state, of poor body condition. Poor body condition also means that they're probably less likely to be able to fight off an infection or fight off a pathogen. And this is even more noteworthy because we now know that striped skunks are coming into contact with other striped skunks and also many other species at these den sites during this period of poor body condition. So that has some pathogen transmission implications. Again, 11 mammal species were observed at these dens, and even though they were not all there at the same time, and I cannot confirm that they were actually denning in the dens, it's a very common practice for an individual, for striped skunks and also other species, to mark territory with feces or urine, which could include den sites. And so consecutive activity, these animals, these different species or different individuals visiting this same shared site where they might be coming into contact with excretions and secretions, urine and feces, for example, we can see some indirect transmission of pathogens, especially in pathogens that might be able to persist in the environment for a period of time. We also noted that correlation between striped skunks and Virginia opossum activity, and it's possible that that could extend beyond activity outside of the den. There's been one study that documented one instance of a collared striped skunk and a collared Virginia opossum being tracked to the same den at the same time. So that could be indicating something greater, but again, with the data that I collected, I'm not able to confirm that. So given all of these pathogen transmission implications, what influences striped skunk denning? So denning duration increased with decreased distance to water. Again, I expect that water is a limiting resource for striped skunks. Denning duration increased with decreased temperature, just again, the lack of ability to find food when it's colder. And denning duration increased with decreased human modification. So in areas where there are humans, striped skunks are in areas where there are human activity. Striped skunks are denning for shorter periods of time. And this is likely because of supplemental food resources. Although human modification did not affect the number of dens that the striped skunks used. This is still really noteworthy though, because another study has documented behavioral changes in striped skunks in areas with human food sources. So what that study found was that striped skunks are more aggressive around certain types of supplemental foods, especially bird seed and cat food. And so if we have these striped skunks that are in this period of poor body condition and coming into contact with all of these different species, either directly or indirectly, and then they're becoming more aggressive, so maybe they're having more direct interactions as well, this really has a lot of pathogen transmission implications for what could be happening here in these urbanized areas. And with that, we're going to move into talking about some pathogen prevalence and spatial and temporal trends in infection. So, as I mentioned, striped skunks are host to many pathogens and parasites that infect other animals, wild and domestic animals, as well as humans. And skunks are also active within this wildlife, livestock, human interface. So this is an interface that is of importance with pathogen studies because we have all of these different host species existing in one area. We know that striped skunks are active in this interface. This is a photo that I captured during this study with some cows just hanging out back here as the striped skunk's out doing his business. And this close interaction also has pathogen transmission implications because, again, pathogens can infect, humans, wild animals, and domestic animals. So as far as striped skunk pathogen analyses, there have been numerous seroprevalence studies that focus on antibodies, so past infections, but there's been little work on active infections in striped skunks or spatial trends that contribute to some of these transmission implications. And no studies have investigated striped skunk pathogen prevalence in the lower Midwest. So the objectives of my study were to determine the prevalence and trends of 5 different pathogens in striped skunks in Southern Illinois. These 5 pathogens fell across several transmission routes. So I was testing for Babesia microti, which is a tick-borne illness. I was testing for Toxoplasma gondii, which is a foodborne illness. I was testing for canine distemper virus, canine parvovirus, and leptospira, which are all fecal urinary to oral route transmission pathogens. I based hypotheses on what we know about these pathogens and how they persist and are transmitted. So toxoplasmosis is often associated with domestic cats, so I expected human modification would have an impact on that. And we know that toxoplasmosis can also persist, especially in water. So I expected water might have an influence on prevalence. Canine distemper virus and canine parvovirus are both associated with domestic dogs. And so again, I expected human modification might have an impact on their prevalence and on infection status in striped skunks. And previous studies in raccoon populations have shown a very cyclical nature in canine distemper virus prevalence. And so I expected some temporal effects on canine distemper virus infection status. Leptospirosis is very common in soil and can easily leach into water as well. And so I expected water might have an influence on leptospirosis infection status. And Babesia microti is a tick-borne illness again. So I expected that land cover and seasons might influence the activity of that tick vector and thus influence the infection status. So for this part of the study, I necropsied 98 striped skunk carcasses that were collected during April of 2018 to March of 2021. During these necropsies, I collected tissue samples and then tested these tissues for pathogens with the Cornell Veterinary Diagnostic Laboratory and the University of Illinois Veterinary Diagnostic Laboratory. So we use different tissues and different test methods just based off of what works best for each pathogen. But it is worth noting here that none of these pathogens were tested in all 98 samples, 98 striped skunk carcasses. And that's because most of our carcasses were roadkills. And I was not able to locate every organ in every carcass because some of them were a little bit flat. But then I also incorporated a spatial and temporal component to these analyses as well. So I again created a 1 kilometer buffer around each collection location to approximate a striped skunk home range. And then I calculated the mean human modification score, that same layer that we looked at earlier, on a scale of 0 to 1 within that buffer. And then I also calculated the percentage forest and field cover within that buffer. And then I calculated the distance to the nearest stream or shoreline from that collection location. I listed the year of the collection, and then I also listed a season of collection. And the seasons were based off of striped skunk activity patterns. So you can see down here, breeding season, young rearing season, winter preparation season, and then winter inactivity season. I built an a priori set of logistic models to test for effects of these predictor variables. So for each pathogen, I built 4 models, one with all predictors, one with spatial predictors, one with temporal predictors, and then one with just those hypothesized predictors from the chart that I showed you a few slides back. And ultimately, I detected 3 of the 5 pathogens that I tested for. So I detected just over 80% prevalence of babesia, about 5% prevalence of canine distemper virus, and just under 30% prevalence of leptospirosis. But I did not detect parvovirus or toxoplasmosis in the striped skunks in the study. When I broke this down by year, I could see that babesia was pretty consistent across all of the years, and leptospirosis was also fairly consistent as far as the level of prevalence across all of these years. But canine distemper virus was only detected in 2 years of the study. So when I took a look at those models, assessing the effects of these spatial and temporal predictors, I had 1 supported model for my canine distemper virus set, and it indicated that only water was significant. So there was a significant positive relationship between the distance to water and the probability of canine distemper virus infection. Leptospirosis was very similar, 1 supported model, and an indication that, again, water has a significant positive relationship, the distance to water and the probability of leptospira infection. And then with the babesiosis models, I had 3 supported models. So I used model averaging to estimate covariate effects and detected no significant effects of any of the covariates on babesiosis. So when I take a look at the prevalence in this study versus the prevalence in prior studies, I run into a few little errors here, or not errors, but issues. The first of which being that I detected about 83% prevalence of Babesia microti in striped skunks in Southern Illinois, but there have been no prevalence studies done in the past for Babesia microti in striped skunks. So there's nothing really to compare that to and what that looks like elsewhere. With Leptospira, I detected just under 30% prevalence, which falls within the range of past prevalence studies. Canine distemper virus, again, just over 5% prevalence, and that is a little bit lower than past prevalence studies. Canine parvovirus, I detected 0% prevalence, which is a little bit lower than past studies. And Toxoplasma gondii, again, 0% prevalence, which falls within the range of past prevalence studies. But you can see here that prevalence in these studies varies greatly, 0 to 71%. And that's because of a few things. Prevalence is really based off of your sample size. So that can have a huge impact on what your prevalence looks like. And also there are different types of tests that can be done. So again, in the past, many of these studies were done based off of seroprevalence, testing for antibodies, and I was testing for active infections. So back to this Babesia microti prevalence, it's extremely high, especially given that I did not expect to detect any Babesia microti because the black-legged tick vector, or the deer tick, as many of you might know it, are pretty rare in the region. And so I really did not expect to detect any Babesia microti. This super high prevalence then indicates that maybe there are additional tick vectors that can carry this pathogen, or maybe there are more black-legged ticks in the region than what was previously expected. But regardless, this is a pathogen that can infect humans. And so this is of human health concern, but of course also of One Health concern. So overall, with the spatial covariates, as distance to water increased, so too did the probability of infection with leptospira or canine distemper virus. And I suspect that this could be an effect of striped skunks using temporary water sources, so puddles and pools that accumulate along roads, because these types of water sources would be less likely to flush pathogens, and they could also accumulate pathogens that are leaching out from the soil. I detected no significant effects of other spatial covariates, but it's worth noting that all of the pathogens in this study have other sympatric hosts. And regardless, this actually has implications for disease transfer, especially in these areas with wildlife and livestock and humans, because striped skunks that are encountered around humans and around livestock are just as likely to be carrying these pathogens that can also infect those humans and livestock. I did not detect any significant temporal effects, although there did appear to be some trend in the canine distemper virus prevalence. Overall, there was such a low prevalence of canine distemper virus that I think it might have just obscured the effects of year especially. And it's again worth noting that several of these pathogens can persist in the environment without a host. And all of the pathogens have other sympatric hosts within the same area that could really muddy any temporal effects that we might have seen. Okay, last chapter. Well, last chapter that I'm gonna bore you with today. So spatial ecology of striped skunks. For this chapter, I focused on home ranges and habitat use. And so a home range is the area that an individual covers while finding food and shelter and a mate. And this can be influenced by a lot of things, including season, sex, age, body condition, and available habitat. So you can imagine the resources that an individual might find in this wooded area with lots of water probably varies a lot from what they're finding in a more agricultural, human impacted landscape. Striped skunk home ranges typically range from about 1 to 3 square kilometers, and they often commonly overlap because striped skunks really are not very territorial or social. When I'm discussing habitat use, I am comparing used versus available habitats at multiple scales. What are they using versus what is there for them to use? Historically, striped skunks have been associated with woodlands and field habitats, but also they exist and persist across this really large geographic range. And so more studies have indicated preferences for prairie and farmland and marsh and edge and industrial areas, and basically you name the cover type and they've been associated with it. So really that just cements this idea that this is really a very generalist mesocarnivore that can persist in many different types of habitats and uses many different food sources. But No studies have investigated striped skunk home range size in the lower Midwest, and only 2 studies have examined trends in striped skunk habitat use in Southern Illinois, and one of them I published a couple of years ago, and it included some of this data. So this is just building upon some of that work. A few studies have also indicated that examining striped skunk trends in habitat structure may be more useful than land cover classes. So instead of talking about do they prefer forest or field, talking about do they prefer a more open canopy and other structural components such as that. So the objectives of this part of my study were to estimate striped skunk home ranges and to examine the effects of sex, season, and habitat on size. Also, I assessed the habitat selection of striped skunks at the home range scale and at the within home range scale, or Johnson's second and third orders of selection. So I expected home ranges would be similar to previous reports, about 1 to 3 square kilometers, with larger home ranges during spring and summer when there's a little bit more activity. Larger home ranges in male skunks because males tend to be a little bit more active, and also larger home ranges in less human modified areas because I expected the individuals would have to travel further to find resources. I expected that habitat use would indicate preference for forested and grassland areas because that's what they've often historically been associated with, and also areas with moderate human modification based off of my previous work, and also based off of the fact that human modification brings with it some supplemental resources. I expected they would prefer areas with low to moderate canopy cover. Even these historical studies out of the 1930s are indicating that striped skunks associated with woodlands are also associated with openings in those woodlands. And I expected striped skunks would prefer areas closer to streams with water as a limiting resource and further from roads because roads can pose a significant mortality source for striped skunks. So for this part of the study, I captured and radio collared striped skunks in the greater Carbondale area during January to April and September to November of 2018 to 2022, and also January to April of 2021. 2020, not 2022, sorry. I tracked animals via radio telemetry at least 3 times a week. And then I built 95% kernel utilization distribution home ranges and 50% KDU core areas. And so what that essentially looks like is if these yellow locations are all known locations of an individual striped skunk, I can use this 95% kernel utilization distribution to estimate the area that the striped skunk is going to spend 95% of its time. So this really encompasses or estimates most of the area that this individual would be using. And then I can sort of narrow that down to a core area, the area where the individual is spending most of their time. And so about 50% of the time, if I wanted to find this individual, I would go to one of these 2 marked areas. But it's worth noting here that we have known locations here outside of this core area, and that's because this is really focused on where the striped skunk spends the majority of their time. So these will come into play as I walk through habitat analysis. For home range analysis, I tested for a difference in home range and core area size by sex with a Mann-Whitney test. I calculated the percentage cover for each habitat class within home ranges and tested for an effect on home range size with a generalized linear model. It's worth noting here that I did reclassify the National Land Cover Database to 5 key habitat classes here, or cover classes: urban, agricultural row crops, pastures and fields, forest and green, and water and blue. I also built seasonal home ranges and core areas based off of those striped skunk activity patterns. And I tested for a difference in size between seasons using a repeated measures ANOVA. For habitat use analysis, I performed home range selection, which is Johnson's second order of selection, where I compared the 95% home range, so that area where they spend 95% of their time, to the available habitat in 3 counties in Southern Illinois. Then I sort of went into a smaller scale analysis here within home range selection, which is Johnson's third order of selection, where I compared the used 50% KUD core areas to the available 95% home ranges. I used chi-squares to test for selection and also for a difference between selection and individuals. And then I used eigenanalysis of selection ratios to really visualize what that selection looked like for all of those individuals. And I did this analysis across 5 sets of habitat variables. I analyzed land cover, canopy cover, human modification, distance to water, and distance to roads. So overall, these are just some of the 95% kernel utilization distribution home ranges of striped skunks. So we see Carbondale up here, and down into here we get into Giant City State Park and the Shawnee National Forest. We've got a lot of ag and ex-urban use on the outskirts of Carbondale as well, and everywhere in between. And over here, we can see our 50% KUD core areas. Again, you can see they're much smaller areas because that's where animal activity was focused. So during this time, I captured 63 striped skunks, and I estimated home ranges and core areas for 41 individuals. That's because I had a cutoff of 30 known locations in order to develop these home ranges. And some striped skunks tended to get hit on the road or met their end in some other various methods soon after they were captured and collared. Home ranges varied a lot. 14 hectares to 1,196 hectares is the range, but the average was about 270 hectares, which is 2.7 square kilometers. So that really is falling within that 1 to 3 square kilometer range that I was expecting. Core areas were smaller, 2.7 to 201 hectares with an average of about 55 hectares. Male home ranges and core areas were significantly larger than female home ranges as expected, but there was no significant effect detected of season or of percentage of habitat cover on home range sizes. Okay, diving into this habitat use, if you're familiar with principal components analysis, these figures are read similar to that. So over here, figures A and B are pertinent to second order or home range selection, and C and D are third order or within home range selection. On the top, A and C, we see our habitat classes, and on the bottom, B and D, we see the individual striped skunks that had significant selection for this set of habitat classes. A larger vector coming from the origin indicates stronger selection, and the direction of the vector indicates which habitat class that individual is selecting for. So in the land cover habitat use categories, we can see that we have this group of individuals that appear to be selecting for developed cover. We have a group over here that appear to be selecting for forested cover, and this group down here that seems to be selecting for grassland types. This is a larger scale home range selection, and we see when we break this down into a smaller scale third order selection, we essentially see this starburst where each individual is really selecting for different things. Okay, there we go. Okay, so with canopy cover, we again see our canopy cover categories up here, 0 to 25%, 26 to 50, so on and so forth. And down here, we can see we had a large group of individuals that were selecting for these lower canopy cover classes with 0 to 50% canopy cover on this side. Over here, we did see some individuals that were selecting for higher levels of canopy cover in that home range analysis, and similar to the last instance, as we move into that smaller scale third order selection, we really see a lot more well, a lot less of a trend. Again, we see there are some trends prevalent in second order selection, but as we move into third order selection, they really start to. So let me see if I can mute. Okay, there we go. Sorry about that, everyone. Okay, as we move into human modification categories, human modification was based on a scale of 0 to 1, with 0 being no human modification and 1 being high levels of human modification. So we can see our categories are broken up here, 0 to 0.25. There's a 0.26 to 0.5 down here, and so on and so forth, all the way up to 1. We can see there are a lot fewer individuals here that had significant selection based off of human modification scores, but we did tend to see a lot more individuals selecting for these moderate levels of human modification versus super low levels of human modification. But again, some variation, and that variation is even more prevalent when we move over here to our third order selection, or that within home range selection. For distance to water, I created rasters with moving windows to really get a good picture of what these individuals' home ranges and core areas really looked like as far as distance to water, and saw that overall there was a very strong trend. It appeared a lot of individuals were selecting for areas closer to water, 0 to 250 meters from water, 251 to 500, and 501 to 750. But there were still a few individuals that were strongly selecting for areas further away from water. And as goes the story, once I took a look at third order selection, there really were no super strong trends prevalent where we had a lot of individuals grouping into one category. And finally, I used a similar method to develop a raster that visualized distance to roads, and detected we had a lot of individuals that were selecting for areas closer to roads, which was not really what I was expecting. But again, as we moved into that third order analysis, we really saw all of the individuals really doing their own things, and less patterns in the habitat use selection. So overall, striped skunk home ranges were extremely variable in this study, and they ranged from super small, similar to those reported in some urban areas, to much larger than average. This large variation may have obscured some of the other trends that I was trying to test for, but it is interesting to see how different those home ranges were within a smaller area. Larger home ranges are common for male striped skunks, so it was not surprising to see that result. And seasonal patterns, I didn't detect any. However, previous studies really varied on whether seasonal patterns were detected or not. So it wasn't necessarily surprising that I did not detect any of those. As far as habitat use goes, this study accounted for variation in selection among individuals, which is unique and hasn't been done in previous studies, but I think is a really important and exciting way to assess habitats, especially with generalist species that aren't all necessarily going to act the same way. We saw some trends evident with that second order home range selection, but third order selection really varied a whole lot, and trends were not really easily detected. And overall, literature shows that striped skunk habitat selection varies really widely across their geographic range as they experience all of these different habitats. But this study was important and interesting because it showed that selection varies widely even at these smaller scales between individuals. And so in summary, striped skunks had really variable habitat preferences even at the smaller scale. They had widespread pathogen prevalence across this urban to rural gradient. And I did detect some winter denning behaviors that changed near urbanization, which could infect or could increase pathogen transmission potential, but also this super variable habitat selection and just the widespread prevalence of pathogens goes into play there too, as far as impacting pathogen transmission potential when we're focused on striped skunks on the landscape. And so big picture, overall, we've detected changes in behavior near urbanization. In this study, I detected changes in denning behavior. Past studies have detected changes in aggression. So what other behaviors change? And do other urban adapted species also experience similar changes? And what does that mean for the community as a whole? I also detected high Babesia microti prevalence. So are there more black-legged ticks than previously thought in this area, or are there other vectors that can transmit this pathogen? Also, how does prevalence in this population of striped skunks compare to populations in other areas of North America where there are known large populations of black-legged ticks, such as the northeastern United States or the northern Midwest? Overall, this comes back to a human health concern, and also from the One Health approach, just a concern in general because it impacts so many things. And I guess leading right into that, using this One Health approach, these striped skunks are extreme generalists that are interacting with humans and domestic animals and wildlife, both directly and indirectly, and they're carrying pathogens that can infect all of the species within all of these groups. And so this just really highlights the importance of continued monitoring and research, because that really promotes the health of all of us. So with that, I'd like to acknowledge lots of folks. My funding sources through IDNR and through the NSF Superb Scholarship Program, my advisors and committee members that have provided lots of guidance over the past couple of years, my awesome field techs and landowners that made all of this possible and tolerated the smell of skunk for many years with me, my supportive colleagues in the CWRL and beyond, and my wonderful family of friends, and Ben and Sadie and Sarah and my mom and dad, and my favorite skunk, Sadie, I already said her. So with that, I will take any questions